Hello everyone. Now that we have a more decent understanding of mitosis, now let's discuss about another kind of cell division which is called meiosis. And in the mitosis video, we simply had a cell which had his 46 number of chromosomes, and when the cell divided, we end up with two cells each having 46 number of chromosomes. So it was it was a type of division in which the number of chromosomes remained equal. Now, what in meiosis, we start off with 46, and we eventually have four cells. We eventually have four cells, each with 23 pairs, 23 sets of chromosomes. So it is a type of division in which the number of chromosomes have been reduced. Now, this type of cell division, the mitosis, occurs in our general body cells, like your skin cell or your eye cell or your intestine cells. Any cell which is your general body cells, mitosis occurs within those cells. And meiosis occurs within a type of cells which are called germ cells. Germ cells. And mitosis occurs in somatic cells. So now we are going to discuss about meiosis, or the division which occurs in the germ cells and which helps the formation of two types of gametes, which are basically sperm and ovum, and each of them have 23 pairs of chromosomes. And so you could imagine if a sperm has 23 and an ovum also has 23, and when they merge together, you end up with 46. So the number of chromosomes in organisms remains equal. Now let's start off with meiosis. First of all, if we have a cell, and just like mitosis, each cell has its interface, which means it has a phase within which it lives its general life, in a phase in which he does his regular things. He grows, he respires, he transports materials within itself. So it's called so this phase in which the cell is just living its day-to-day routine and doing all the things which he does, it's called the interface. And the interface, as we know, has been divided into a G1 phase, an S phase, and a G2 phase. So even in whether we are talking about meiosis or mitosis, they are both the same till here, and later on there's a little difference. So in the S phase, when it reaches the S phase of its interface, the S phase, the number of the chromosomes, or the chromatin, which is again a chromatin is just a DNA. Chromatin is just DNA wrapped around proteins, which are called histones. So now in its interface, the genetic material of the cell is in chromatin form. It's sort of jumbled into each other, and during the S phase, it replicates. The chromatin, like if it has one, it replicates into two. And we discussed in a couple of videos back that how does the DNA actually replicate itself, and it's quite an amazing process. But the point is, in the S phase, the DNA just replicates itself. Now, further moving on, after it goes from the S phase, the DNA, the genetic material replicates, and it's still in this coiled form. We can't really distinguish it. It then goes to the G phase, the G2 phase, and it's the second gap. G basically, I think, stands for gap, first gap and second gap. So in the G2 phase, it also lives its day-to-day life and just regular living of the cell. Now, when it enters, the first stage is called the prophase. Prophase 1, actually, because meiosis is also of two types. We have meiosis 1 and meiosis 2. So actually, so this is really actually prophase 1. So in prophase 1, what happens is that the uh, what happens is that the cell, and within the cell, the genetic material or the chromatin condenses, or it wraps itself around enough, or it becomes like this, like an X-shaped structure. So it becomes visible. It becomes visible, and at the same time, the nuclear membrane around this starts to disappear. So the nuclear membrane, so this nuclear membrane starts to disappear. And the two the centrioles, which are the microtubule organizing centers, start to move in the opposite direction and try to form spindle fibers, which help. So these are the early spindle fiber formations right here, which help to which later on will help the movement of direction of the chromosomes. So after prophase 1, we have metaphase 1. Metaphase 1. Now, the key thing which I wanted to mention is that when we are talking about prophase, just simply in mitosis, the chromosomes just replicate and they condense and we see them. But when we are talking about prophase 1 in meiosis, especially in meiosis, when the chromosomes replicate, this chromosome which replicated tries to find its homologous pair. Homologous pair. So you might be thinking, what is a homologous pair? Well, as I mentioned before, a cell has 46 sets of chromosomes. Split it in half. You have 23 and 23, right? So this 23 sets of chromosomes has been gained by the cell from its father, and this one has been gained from his mother. So now, so you might be thinking, so now think for a second. A chromosome which has a gene right here, let's say he has a gene for black hair, and this one is, let's say it's from your father, and this chromosome, which is the other chromosome, these, like one of this 23 was this from father, and this one of this from the 23 is from the mother. So this one also has a part of the gene which encodes for the same kind of trait, which means hair color, but this one, instead of black, is blonde hair. So you see that both chromosomes encode for the same kind of gene, but they have the different versions of a gene, or in other words, they have different alleles of the gene. So a homologous pair of chromosomes, essentially a chromosome which pairs of chromosomes which have been inherited from your mother and from your father, but they encode for the same gene too, but they have different versions of the gene. So when in prophase 1, when the chromosomes, in prophase 1, when actually the chromosomes replicate, they try to find their homologous pair. So if I were to draw just the nucleus of this cell right here, just the nucleus of it, and let's just, for the sake of simplicity, let's say this organism has 4 sets of chromosomes, and I would like to draw them in 4 different colors, so it would be easier. So this is another one. So this is another one, and this is another one. So let's just say these 4, let's just say it's a cell which has 4 sets of chromosomes in it, and it when it replicated, it tried to find his homologous pair. So the green's homologous pair is the blue one, and the purple one is the yellow one. Now what happens is that when it finds its homologous pair, what it does is that it switches up the place. Now I would like to show you this. This is the chromosome, and this is another chromosome. So let me draw it here. So you see, this part of the chromosome, let's just say encodes for eye color. Let's just say it's black, and this part of the chromosome has encodes for also for eye color, but it's for blue. But so what happens is that when it finds its homologous pair, they come close to each other. They come so close that actually they overlap. So if I were to draw this chromosome, and the purple one would be something like this. So they overlap with each other. So now you have a combination of 4 chromatids, and by chromatids I simply mean that if this is a chromosome, its individual arm is called a chromatid, and when they are separated, so if I separate these two, they again become chromosome. Now, so hopefully I'm not confusing you. When they are individually, they are called chromosome. Combiningly, the whole structure is also called chromosome. But when they are combined, the individual part of it is called chromatid. So now, since they are combined, there are 4 chromatids, and with the 4 chromatids, we sometimes, no, not sometimes, they're actually called the tetrads. This 4 combination is called the tetrad. Now, what happens after this process, when they come close to each other? So let's just say this part right here encodes for blue eyes, and this part right here encodes for brown eyes. So what they do is they switch to, they code for the same gene but different versions of the gene. Keep in mind that. So what they do is they just switch their places around. They switch the genes around. Then this process is actually called the synapses. Synapses. In a process in which these two, the alleles or the versions of the gene, switch their places around. So now you basically have on the yellow one you have a little bit of, so on the yellow one you have this purple. So if I were to draw this here, on the yellow one you have this purple, and on the purple one you have this yellow one, right? So this is what in prophase, which is one of the most important one of the most important stages of the meiosis 1, is this is it. This is what happens in prophase 1, and same goes with these 2 chromosomes right here. They just do from the process of synapses. They you might want to view them as that they switch their genes around. Now, so let me remove this. Right. Alright. So now after prophase, you have metaphase. And what happens in metaphase is that, now actually I will just draw the cytoplasm, since the nuclear membrane has disappeared. So what happens is that in metaphase you have these centrioles which have microtubules emerging out of them. So these microtubules like this, right? So these microtubules are fully formed, and let's just cut this part. Cut, paste. So let me erase that. So these microtubules have uh, fully formed, and these chromosomes have attached themselves to these microtubules. Now, what happens is that actually when these microtubules starts to pull apart the chromosomes, and what they do is that instead of pulling like in mitosis, instead of separating the chromosomes from their centromeres, what they do is that they actually pull the whole chromosome apart from its homologous pair. So if I were to draw this, let's just say this is a chromosome, and this is its homologous pair. This is another chromosome, and this is its homologous pair. So if the microtubule wants to pull it, so what it does is that it pulls these two chromosomes in the opposite direction, and it pulls these two chromosomes in opposite direction. And if you notice, you start off with 4. I forgot you have to draw it 4 here. So you start off with 4, and their homologous pairs, and when you separated them, you end up with 2, right? So in the metaphase, they align, and instead of the microtubules pulling these two apart, they actually pull the whole thing from its homologous pair. So in prophase, metaphase. So this is what metaphase is all about. So after metaphase, you have prophase, metaphase, anaphase. In anaphase, what happens is that they keep pulling themselves apart, and they keep pulling themselves apart. Eventually the cell structure becomes something like this. Well, not like this. It's actually, let me remove that. The cell structure actually becomes like elongated. It elongates. The cell elongates a little bit, and the microtubules right here. So I just rotated this structure right here. So this it's in the horizontal. I just made it vertical. So please don't get confused. So now you have these 4 sets of chromosomes. I mean 2 here and 2 here. Now, so in anaphase, they just pull each themselves apart. The microtubules are pulling them apart, and these are the microtubules pulling them apart. And this is these indentations which are starting to form are called the cleavage furrow. And as it happens, when they are on the opposite sides, the nuclear membrane starts to reappear around them, and cytokinesis starts occurring place. And cytokinesis means division of the cytoplasm. So anaphase and cytokinesis occur simultaneously. So they occur simultaneously, and so at the end of anaphase 1, so this is metaphase 1 and this is anaphase 1. So at the end of anaphase 1, you end up with two cells, they're chromosomes, and two chromosomes with attached like they have a chromosome with two attached chromatids in them. So you start off with 4 right over here. Then here is this 1, 2, 3, 4. You detach themselves. So now you have 2. Now the meiosis 1 is finished. Now we are ready to enter meiosis 2, and meiosis 2 is identical to mitosis, because now the same process occurs. The same kind of process occurs within meiosis 2, And the meiosis 2 first starts with um, prophase 2, in which first of all, when meiosis 1 is finished, the cell again goes in the interphase where it unwounds its chromosomes and it lives its life. But when it enters meiosis 2, it recoils again, it condenses its genetic material again and become in this X-shaped form. So in meiosis 2, when you have prophase 2, and what happens in prophase actually is that the same thing, the genetic material starts to disappear, and what happens is that the genetic material, when it disappears, you not the genetic material, the nuclear membrane starts to disappear, and the genetic material becomes condensed, and the microtubule starts forming around the cell. So if I were to draw a cell, and here is the nuclear membrane. So the genetic material becomes condensed, and the microtubules here, the centrosomes here starts to form early formation of the spindle fibers. And again, in prophase and metaphase 2, what happens is that the chromosome starts to align. So now you have this huge cell, and you have these centrosomes on the opposite side, and you have these microtubules, and so let me draw this. So now these chromosomes right here, so let me draw it in another color, this yellow. So now these chromosomes align. Now what happens is that actually, so the chromosomes actually, let me remove this. So the chromosomes actually so the chromosomes actually align themselves like this. I picked the wrong color. I drew them like this for a purpose, because and it has to be in the center. Has to be about in the center. It's about in the center. Now what happens is that now in metaphase 2, instead of pulling the whole chromosome again, what it does is that it splits the chromosomes into two chromatids from its centromere. So this part right here goes in this direction, and these two parts goes in the opposite direction. So after metaphase 2, we have anaphase 2, as well as cytokinesis occurs at the same time. So in anaphase 2, what happens is that let's just say the cell, so these are the microtubules, these are the microtubules, and this is the whole cell. So now the cell is starting to elongate as these two chromosomes start to split apart. And how they split apart is that, so this part moves here and this part moves here. So now you can imagine when these two are separated, they're individually, now they're again called just chromosomes, simple chromosomes. And here, when they were attached to each other in this cell, in these two cells, we still had two, but with 4 chromatids. Now, when these chromatids split apart, we eventually end up with again 2 chromosomes. So now what happens is that in metaphase 2, in anaphase 2, these two get pulled apart along with cytokinesis occurs, and here you have these indentations where the cytoplasm starts dividing up, and then you end up with two cells, each having 2 chromosomes. So, but this is only one cell we're talking about. We actually had another cell which was going through the same process, right? This one, we just picked up this one. This one is going through the same process again. So you have 2 more cells. 2 more cells which have 2 sets of chromosomes. So you start off with, all the way here, you start off with 1, 2, 3, 4 in the cell. You start off with 4 in the cell, and we end up with 1, 2, 3, 4, 5, 6, 7, 8 chromosomes. 8 chromosomes. Imagine if you look, we have 1, 2, 3, 4, 5, 6, 7, 8 chromatids. So if individual chromatids are also called chromosomes. So we have 1, 2, 3, 4, 5, 6, 7, 8, 4 cells with each with two kinds of chromosomes, half of the number of the original cell. And these kind of division helps actually in formation of gametes such as sperm and ova, which helps. So so let's just say, like in another organism, meiosis has been occurred. So if this is the sperm and this is the ova, it also has 2. The sperm also has 2. When they join up, you again end up with 4. So it helps to keep the number of chromosomes equal in organisms. And this variation, which this switching of genes, versions of genes on the other homologous chromosome, this actually helps in variation, which hopefully will be my next video on, in which we'll just go in a little bit of detail and try to understand it. And actually I actually downloaded a picture of, oh, here it is. I actually downloaded a picture to so if you guys have a little bit of confusion about the drawing which I drew. So you see, like 1, 2, 3, 4, we have 4 chromosomes. They replicated 1, 2, 3, 4, and still we have 4 chromosomes. Because if you're talking whole, these are still chromosomes. So this is a really important point to remember. So during meiosis, chromosomes from both parents are copied and paired to exchange portions of DNA. They say paired, so they pair with their homologous pair. So you see the yellow, so this blue one pairs with the red one, and this blue one pairs with the red one. They both encode for the same kind of gene, but different versions of the gene. And what they do is that they switch a little bit of sections of the DNA or parts of the DNA on the other DNA. So you see that this blue one has a little bit of this red one, and this red one has this blue one. So now they divide up into 2, 1, 2, 1, 2, 2 chromosomes. And these two, so the process in which we get these 2, this whole process is called meiosis 1, till we reach these 2. And from these 2, like 1, 2, these 2 divide, and these 2 chromosomes are separated from each other, and these 2 are separated, and one of them goes here, 2 of them goes here, 2 of them goes here, and this cell also divides, and we end up with 4. So if you notice something, if you go all the way back, we have 1, 2, 3, 4, and we have 1, 2, 3, 4. So the original cell had 4, this cell had 2, this cell also has 2, the half of which the original parent cell had. So this was meiosis 2. So hope you found that useful, and I'll see you in the next video. Bye bye.